Another video for A-level biology. This is video 2.3, I believe it is, which is analysis of cell components. Now, what that effectively means is it's looking at how we study cells, basically the microscope stuff, and then how we prepare samples. So what do we need to know? Well, if you look at this picture, these two pictures here in the green show you pretty much what we're going to start off with. So here we've got a chloroplast, extremely small version here, and a much larger version here. So that would be magnification. It's how many times bigger the image is compared to the object. But if you look at this one, it's less possibly clear. And maybe I'm saying clear, I'm actually dropping you a hint, because that's to do with the resolution. So what we're going to look at is, first of all, magnification and resolution. So magnification is the number of times larger an image is compared to the real size of the object. So you've got the equation there: magnification equals size of object divided by size of image, divided by actual. Okay, and we'll talk about that in a few moments, how to remember that. The resolution, you could say how clear an object is, or the clarity, but unlikely to get the marks at A level for that. So for resolution, what you would say is it's the ability to distinguish two objects that are close together as separate objects rather than seeing them as one blurred together. So let's have a look. So before we look at magnification, we need to introduce you these different sizes that we may not have looked at before. So effectively, what you should hopefully be aware of is you have centimeters, okay, and then the smallest thing on most rulers would be 1 millimeter. Now 1 millimeter has 1,000 micrometers in it. So if I wanted to know, let's say I had a object that was 3 millimeters in length, and I wanted to know, well, how many micrometers is that? Because there's 1,000 micrometers in every millimeter, I multiply that 3 millimeters by 1,000, and I would have 3,000 micrometers. Okay, so you always multiply the millimeters by 1,000 to get micrometers, and that is the average cell is somewhere between 10 and probably 50 micrometers in size. Want to go smaller than that, study viruses, we need to go down to nanometer scales. So nanometers is 1,000 micrometers. So if I've got 3,000 micrometers, that would then, I would have to then times that again by 1,000 to work out how many nanometers it is. So 3,000 would become 3 million nanometers. And then you can get even smaller than that, but you probably won't need that in A-level biology. But as we get down to this bottom scale, we're probably talking about the structure of DNA and things like that. So then, first of all, what is resolution? We said the resolution is this ability to distinguish two very close objects as separate objects, so they don't just blur up. And basically, a lot of microscopes are actually more limited possibly on the resolution than the magnification. So the resolution of the human eye is 100 micrometers. So that means anything smaller than 100 micrometers apart from each other, anything closer than 100 micrometers apart from each other, or anything smaller than 100 micrometers, we would struggle seeing. The light microscope takes it up to 0.2 micrometers or 200 nanometers, and the electron microscope takes it up to 0.0002 micrometers. So you can see the importance and resolution of these microscopes. And now we've got a light microscope, or some people would call it an optical microscope, and you'd be expected to be able to talk your way through setting an image up on here. So we'll talk about a bit later about putting samples on slides. But if you've got your slide, you'd put your slide onto the stage at the microscope. You would then look through the eyepiece lens, which is generally ×10 magnification, and you would put the smallest objective lens on to view the image. If that was ×4 ×10, which is your eyepiece, would equal ×40 magnification. You then use the coarse adjustment focus, and that focuses the image so you can just about see it. You then use the fine adjustment focus, and then you increase the magnification by changing the objective lens if needed. But if you want to look at that, which you do need to know, I'd suggest looking at the textbooks. Not very difficult, you just need to look at it and make sure you understand it. So this is the sort of thing you'd see from an animal cell under an optical microscope lens. So they've got a limited magnification about ×1,500. They've got that resolution we just looked at of 0.2 micrometers, So you are limited. You can see here, you can see the nucleus of the cell. So this is a micrograph of it here, and you can see some darker patches. But really, the resolution is very poor. You can't distinguish any of these structures that we're sort of looking at. And the same for a plant cell. You can see the chloroplast really clearly, the cell wall really clearly. But other than that, it doesn't look like there's a lot in the actual cell, and that's why we need to use more higher magnification, high resolution microscope. So we need to be able to work out magnification using the equation. We need to be able to rearrange the equation. So I always use the IAM equation. So the I stands for the size of the image, the A stands for the size of the actual, and the M stands for the size of the magnification. So I can work out image size by doing actual multiplied by magnification. I can work out actual by doing image size divided by magnification, or I can work out magnification by doing image divided by actual. So, and the only thing other is than that is basically learning it and being able to convert maybe micrometers into millimeters. I'll just show you why now. So here's an example of a question you might get in an exam. And if it says image size, work out the actual size. So that's what we're working out in this experiment. What is the actual size of this cell? So we know that the image is 50 millimeters. And we're going to divide that by 1200, which is the magnification. And the symbol for magnification, by the way, the units for magnification, just put a multiplication sign in there in front of it. Okay, when we do that, we do 50 divided by that, it equals 0.0416 millimeters. But wait, when have you ever calculated the size of a cell in millimeters? So what we need to do is we need to convert the millimeters into micrometers. So instead of 50 divided by 1200, we're going to do 50,000, because in 50 millimeters there are 50,000 micrometers. Remember, every millimeter has 1 micrometer. And that gives us a bit more of a realistic answer. Remember earlier on we said that the average size of a cell is between 0 and 50 micrometers. Well, that would give us an answer of 41.6 micrometers. I'm struggling to draw on there for you with one hand. 41.7, I suppose, if you round it up. Now, you could have done that a different way. You could have worked this out and then times it by 1,000 to convert it. But I always prefer to turn my millimeters into micrometers to start off with, so I don't forget to do it later on. And that is actually quite a common error that people do forget to do it later on when they're working out their answer. Okay, so then I want to look at higher things under higher magnification, higher resolution, because I need to see more detail. So what I use is an electron microscope. An electron microscope, basically, just you only need to know really basically how it works, is it fires a beam of electrons down a vacuum. It has to be a vacuum because it stops distortion of the electrons. The electrons then hit a sample which is at the bottom of the tube, and then depending on what type of electron microscope it is, they project the electrons onto a screen, produces a black and white image called an electron micrograph, which is what we've looked at before. We've looked at pictures of cells. So there are two types of electron microscope. The first type is called a transmission electron microscope. Now this is slightly less powerful slightly more powerful in magnification, and it has times 500,000 magnification, so extremely high magnification. And what this is usually useful for is for providing images of 2D images of the cell. So this is where your typical picture of a cell, where you can see the nucleus, the chromosomes, the mitochondria, the ribosomes, whatever. That is being formed using a transmission electron microscope. It transmits electrons through your sample, so they pass through the sample. So imagine that your sample is here, okay, on the transmission microscope. The electrons pass through it, and it gives you a 2D image of whatever you're looking at. So it's a good way of mapping out a cell, for example. A scanning electron microscope is different. It's got a much lower magnification, 5 times lower, but it's useful because it gives us 3D images. Now, why is that potentially useful? Because the third dimension, as you should know, is depth. Okay, so what that gives us is it allows lots of surface details. So if we want to look at the surface of, let's say, a virus, and we want to look at the viral, like the integrity of the virus, the outside of the virus, we could look at that. We want to look at the surface membrane of a cell, we could look at that. So it gives us this 3D shape. Both of them produce, however, 3D but in less detail. Both of them produce black and white images. So if we're looking at specimens, how will we prepare a specimen for this? Specimens must be extremely, extremely thin for electron microscopes. They need stain so we can see different parts. Think of an X-ray. Basically, the way that they work is like an X-ray, apart from instead of the X-rays passing through your soft skin and not your bones, you stain the microscope using heavy metals, normally like uranium and lead, but gold's also used. And that will get absorbed. So let's say they get absorbed into the nucleus, into the chromosomes. Then that means the electrons will not be able to pass through the chromosomes, and you get a whiter image there. And as a result, you get the different contrasts of dark areas. So the ones that prevent the electrons getting through get the dark areas. There are problems with electron microscopes though, and they must be in a vacuum, because otherwise the images get distorted because the electrons would be just slightly altered in their course by the air in the microscope. You cannot look at living specimens because, as we said, you have to prepare it in a vacuum. Because it's in a vacuum, a lot of the water is removed from the cell because of the low pressure that it creates, and therefore the organism will always certainly die. Also, the water itself would distort it, so it causes a problem. So as you know, water makes up such a large amount of living things, it wouldn't work. Another disadvantage is black and white images. So the image and colour that you see, the red blood cells, they would look like the first black and white slide to begin with. We add that colour on called false colour using computer programs, software programs, because we want to see things in the colour we expect to see them. But it is only black and white, so we're not seeing the natural colour when we're looking under an electron microscope, whereas under a light microscope you can. In terms of preparing the microscope slide, obviously this will be much more for your controlled assessments, your PAGs, or your required practicals, depending on what course you're doing. They basically, you do this sort of stages here. The first thing you do is you should always add a droplet of water onto the slide before you add anything on. You then place your specimen into that droplet of water to keep it in position where you want it. You might then add a drop of stain onto there because you need to provide that contrast in the cell. You then place a cover slip gently over the edge and slowly press down on it to remove any air bubbles. But wherever you do any microscope work, you are inevitably going to see artifacts. Now think back to when you were in like year 7 and the teacher first did microscope slides with you. You probably looked and went, oh, I can see the cell, I can see the cell, but it was probably an air bubble. So an air bubble is a good example of an artifact. It's something that you think might be part of the cell, but it isn't part of the cell, and you can see it there. So it might be air bubbles, might be dusts, it might be from how you've squashed or stained it, that you might have inaccuracies there. So it might be when you've squashed a bit of onion slide, it's folded over itself, and you think it looks like something that's important, but it isn't. And basically, the more preparation a sample needs, the more artifacts you tend to get. So electron microscope slides samples tend to have a lot of artifacts. And the only way you can tell for certain is it an artifact or not is by redoing the method slightly different and seeing have you still got the same things that you're seeing there in the first place. If you don't, then it's most likely to be an artifact, not a part of the cell. And the last one is, how do we actually study cells? How do we get the organelles out? So we can look obviously whole cells, but we might want to look at the nucleus, we might look at the chloroplast, we might want to look at the mitochondria. Well, how we do this is a several stage practical really. So what you would do first of all is something called homogenization, which is basically breaking up the plasma membrane, so to get the contents of the cell out. So we break up the plasma membrane, so you'd use just a blender or something like that to blend up the cell membrane, to break the plasma membrane open. You would then extract what we call the homogenate from that and filter it to get rid of like the big bits of plasma and the debris of the cell. Then if we're going to store the contents of the cell, we'd store them in a solution that is ice cold. When that is to stop enzymes from continuing to maybe break the cell down. We'd store it something called isotonic, which means that osmosis wouldn't have an effect, so the cells wouldn't get damaged. And we'd store it at buffered pH, so we don't denature the enzymes. We'd filter it, as I just said a moment ago, which would leave the organelles, and then you'd put it in an ultrafiltration centrifuge, basically. Sorry, an ultracentrifuge, not ultrafiltration. And the centrifuge, basically, you'd start off with low speeds, and you'd take that and you would spin it in a centrifuge What that would do is it forces the mass, the things with the heaviest mass, to the bottom of the tube. So at the bottom of the first tube at low speeds, you would get a pellet, which is like a dense bit of cell basically, which would be the nucleus. We then take out the supernatant. Now, the supernatant is the liquid that's still floating on top, which would be a mixture of the lighter organelles in the cytoplasm, and we put that into another test tube. We would spin that so you get another pellet at the bottom of dense material. So we spin it at a slightly faster speed this time, and you would get the next smallest thing down after the nucleus, which would be the mitochondria. We then get the cell contents out, the organelles out, the homogenate out, and we would spin them again at a faster speed, and that would get things like microsomes out, bits of the plasma membrane out, and then again we'd spin it again, and then we'd be starting to get to the real small contents of the cells here, so you'd get a pellet of ribosomes. So you might have to do it, always start at lower speeds to get the bigger molecules out, the more dense organelles out, and then you slowly increase your speed and you get to the lighter organelles. So often in an exam, they'll ask you, how would you get ribosomes out? And you've got to know the order. So it goes from a nucleus to a mitochondria to lysosomes to the ribosomes. So there you go. I hope it's been useful, guys. I'll see you next time.